So thank you so much, Alessandra, for my introduction. I will present today this project made in Trondheim, in the Norwegian University of Science and Technology. I'm from the University of Las Palmas de Gran Canaria in the Canary Islands in Spain. So today, the lumpfish industry has a challenge, with this that the lumpfish production is currently characterized by variable growth and survival during the larval stage. So here, the industry needs a better know of the nutritional requirement, a better know of the functional behavior of lumpfish larvae, and as well how larval and juvenile behavior responses are affected by the nutritional quality of the first feed. What is the solution to the lumpfish industry need? So we found that Artemia and Cirripedia were the best solution for this, for improve the start feeding of this aquaculture species. Artemia provide a significant better growth and survival, as you can see here in these graphics. The mortality and the standard length of lumpfish larvae were improved when we feeding Artemia. It was the best treatment for the standard length and as well for the mortality, followed by Cirripedia. However, when we were studying the feeding behavior of this species, we found that when we feeding lumpfish larvae Cirripedia, they provide, this treatment provide a higher number of successful attacks, and we saw significant difference, and as well Cirripedia gave us more larvae activity measured by time swimming. So how we got this? We used five different diet treatments. We use commercial dry feed, enrichment Artemia, copepose, Cirripedia, and as well a combination of copepose and Cirripedia. It was made in an acclimatized room using automatic feeders, 10 degrees, and with a photo period of 24 lights in 100 liters tanks, and we use a replicate. So in total, we got 15 tanks, and we studied this lumpfish larvae until 33 days post-hatching. So to record or to see the feeding behavior, we sampling the lumpfish larvae at 16, 22, and 28 days post-hatching. We used 10 larvae per tank, and they were sampled for each tank and then placed into 1 liter plastic bottles. For 3 hours, larvae were not feeding, and they were acclimatized. After these 3 hours, we add a volume of larvae of 350 Cirripedia per milliliters plus 350 Artemia now applied. Then the larval behavior was recorded for 2 minutes. The parameters that we check and we measure was the time that the larvae was swimming, the time that the larvae was attached to a surface, and the time that the larvae was orientated in that bottle. As well, we measure the number of successful and unsuccessful attacks, and as well realized that the larvae were doing and display different types of attacks, swimming or attacks. As you can see here in this video, and sorry, it's not too big, but at least you can guess. Sorry. Here. That's the swimming attack. The white spot that you can see is the larvae. So here we go. And then the second type of tax. Here we go. So who are the potential applicants that the industry or end users will have when you for this study? So first, lumpfish, of course, lumpfish hatchery and the lumpfish industry, aquaculture feed producers, and as well academia, lumpfish aquaculture research. So how this end user could use this output? So for example, the lumpfish hatchery and industry optimize the start feeding protocols for this species. The aquaculture feed producer could improve the quality and robustness of this lumpfish, of this lumpfish larvae, and obtain or get high quality lumpfish, which could control better the sea lice when they will transfer to the sea cases. And finally, lumpfish aquaculture researchers continue researching the key areas in the production cycle of the lumpfish, which are so many challenges today. What will be the impact when the end users apply this output, our output? So when feeding lumpfish larvae artemia, it increases the lumpfish larva growth, which will increase the overall production and will have a more economically beneficial than the normally the often treatment to feed this lumpfish in commercial hatcheries. It's better, a more economical benefit than using dry feed. And if they use Cyclopterus, we show that increase the pre-attack success, and it will be a better larval and juvenile quality, which may result in a higher capacity to eat the sea lice in the future when we'll transfer the lumpfish juveniles to the sea cages. Now, this technology has validated in the lab, and the next steps are to a presentation, a poster presentation next week in Madeira, in Portugal, in the Aquaculture Europe 21. But we need to link these results with the nutrient content, especially the lipids and protein content in diet. We couldn't do it due to COVID. We have restrictions, and when we will these results, we will publish in peer journal. So thank you so much for this opportunity to show you our results. You can contact me by email or LinkedIn, and I will have to say that thank you so much for the people was involved in this research. It was impossible without them. So thank you so much.